So I want to first start out by giving a little bit of a primer on virology, and this will be Virology 101, really just focusing on some of the important characteristics that help you understand how this virus works. So viruses are generally classified according to their genomic characteristics. They're either RNA viruses or they're DNA viruses. That RNA or DNA is either single or double-stranded. It's either, in the case of RNA, positive or negative sense, and it can be segmented or just one whole piece of RNA or DNA. And then we also classify viruses as to whether they're enveloped, which they generally pick up when they bite off a whole cell, or non-enveloped. So in the case of the coronaviruses, they're single-stranded. That is, they're RNA, they're made of RNA. They're single-stranded. It's positive sense, meaning it's directly translatable into protein. The RNA strand is non-segmented. In fact, it's very long. It's the longest genome of any RNA virus. And finally, these viruses are enveloped. And this is just a schematic of the virus. The important features of a coronavirus are, first of all, the spike protein, which is here in orange. And that's responsible for the crown that you see on electron microscopy, which I'll show you in a bit. And of course, gives these viruses their name, coronavirus. Deep to the spike protein is the membrane glycoprotein, which forms a capsule around the nucleocapsid protein, and that in turn covers the viral RNA. The complex between the nucleocapsid and the viral RNA is shown in this thing that looks like a small intestine. And here is an electron microscopic picture of the virus, and I think you get a very nice idea of why these are called coronaviruses. So I want to spend just a bit of time on virus replication. This turns out to be a rather complicated affair in these viruses. I'm going to keep it simple and really focus on just those aspects in viral replication that are important in terms of immune responses that might be protective, or in terms of targets for antivirals. So the first thing that has to happen with this virus, which we call SARS CoV-2, by the way, that's the cause of COVID. But both SARS, the original SARS CoV virus, and SARS CoV-2, both have to attach to their host cell. And this attachment is done because of an interaction between the spike protein on the virus and ACE2 on host cells that are susceptible to infection. ACE2 is angiotensin converting.
We're learning about the virus.
Enzyme 2, and that is a protein that's expressed on many cells, but importantly for our discussion today, is expressed on cells in both the upper and the lower airway. So binding of the virus to its host cell is a critical point in viral replication, and it is very frequently a target for antibodies, because if antibodies can interact with the spike protein in such a way that they inhibit the binding of the virus to its host cell, you won't get infection. And in fact, you'll get neutralization of the virus. So one can imagine then that in developing a vaccine, one would want to mount antibodies that bind to the receptor binding domain in the spike protein, so that binding of that protein can't occur with its receptor ACE2. We think that's going to be a long time before we get that vaccine, but another approach that can be effective at this very early stage is to passively infuse antibodies, either antibodies that are in the plasma of a recovering patient, or monoclonal antibodies that are generated in the laboratory. And again, by the passive infusion of these antibodies, you can inhibit the binding of the virus to its host cell and completely neutralize virus infection. But if the virus does bind, the next thing that happens is it gets internalized into an endosome. And unfortunately, I don't have a pointer to show you, but I may be able to do this with a cursor. And so here is the endosome. The virus has gotten now into this endosome. And what happens there is that the virus membrane will fuse with the endosomal membrane. And when that happens, the viral RNA exits the endosome and winds up in the cytoplasm. Now, the important part about this step within the endosome is that this is where we think that drugs such as chloroquine and hydroxychloroquine work. We don't know exactly how, but at least it's entirely possible that they work at the step of endocytosis and inhibit the ability of the virus to fuse with the endosomal membrane. Furthermore, antibodies, again developed either with a vaccine or infused passively, can potentially inhibit this fusion process, again stopping viral replication. But if that viral RNA does get into the cytoplasm, the next thing that happens is this viral RNA is translated into a big protein, which is called a polyprotein. This polyprotein then has to be chopped up into multiple small, what we call non-structural proteins that play a major role in subsequent steps in viral replication. In particular, they're involved in replication of the viral genome. They're involved also in tamping down immune response, the host defense to this virus. But in order for that polyprotein to be cleaved into these smaller non-structural proteins, the virus requires a protease. And it makes a couple of proteases. And again, this is an important potential target for drug development. Because if we can inhibit that protease, then this polyprotein just stays as one big protein, doesn't get chopped up, and the rest of replication cannot occur. And this is a particularly good target because we know that protease targeting works very well in treating infections such as HIV and hepatitis C viruses. The next step in viral replication is transcription of the viral genome. So this RNA has to make copies of itself in order to eventually be packaged in the virus. And this is, again, a very complicated process in this virus. But the most important point is to realize that one of the enzymes that's required for transcription of the RNA is the RNA-dependent RNA polymerase of the virus. And this becomes another key target for drug development. And in fact, one of the drugs that's being tested here at UCI, as well as in many other places, remdesivir, is exactly the kind of drug that inhibits the RNA-dependent RNA polymerase of this virus, as well as a number of other viruses. So eventually, though, if there is no treatment, you have the virus proteins being made, you have the virus genome being replicated. All these things get packaged together, and yada, yada, yada. You have a new viral particle. That viral particle then egresses from the cell in a vesicle and eventually winds up outside of the cell where it can infect a new cell or a new human. And again, this egress of the viral particle out of the cell is another potential step in therapeutics. It's thought that chloroquine and hydroxychloroquine may also affect this step in viral replication. Okay. Which coronaviruses infect humans? Well, there are 4 common ones that have been circulating, we've known about for a fair amount of time. And these merely cause common colds, although they can cause lower respiratory infections, sometimes severe, but almost exclusively in immunocompromised patients. But then we have 3 bad ones. We have the agent of SARS, which is SARS CoV. We have the agent of COVID-19, which we're talking about today, which is SARS CoV-2. And then we have MERS-CoV, Middle Eastern respiratory syndrome virus. There's no question that SARS COV-2, the cause of COVID-19, is a new virus. It simply has not appeared before. If that were the case, we would probably not be seeing what we see now with this explosion of an epidemic. It most likely came from bats, and I'll show you the reason that that's thought to be the case in a minute. We do not know if it spread directly from bats to humans or if there was some intermediate animal that served as a host. We do know that the epidemic was first identified in association with the seafood and live animal market in Wuhan, China, but we really don't know if the first cases occurred at that market and whether that just happened to be where the first cases showed up, as opposed to some animal in that live market being involved. We simply do not know the answer to that question. What I'm showing here is the horseshoe bat, the Chinese horseshoe bat. It's a very lovely creature, as you can see. And this harbors organisms, coronavirus is very, very similar to CoV-2. And here on this phylogenetic tree, we can see how these viruses are related to each other. So in red are the COVID viruses here. Whoops. And you can see these are just different isolates of the same virus, really very, very similar, maybe one or two amino acids different. This green one here was isolated from a horseshoe bat. You can see it's very, very closely related to SARS CoV-2. And then there's a pangolin virus isolated from pangolins that's also quite similar to SARS CoV-2. Way up here is SARS CoV, the agent of SARS, and that actually turns out to be quite similar to SARS CoV-2, sharing about 82% homology with the new virus. Down here, also a little bit more distant, is MERS-CoV virus. And then down here are the common circulating human cold coronaviruses, these four down here. So a question that we're often asked is, are coronaviruses seasonal? And if so, can we expect COVID to be a seasonal disease? And I guess in some ways, most of us are hoping that that's the case, because then we'll have some relief over the summer. But this is really a dual-edged sword, because if it is seasonal, I think one might predict that it'll come back next winter, and the winter after that, and the winter after that. But in any case, one thing we know for sure is that the common circulating human coronaviruses are quite seasonal in their ability to attack human beings. So what you see here is the line that represents influenza frequency over 3 years, 2007, 2008, 2009. And you can see that, as we all know, influenza activity peaks in the winter months, January, February. What we also see here in the bars are the other human coronaviruses that cause common colds. And you can see that they really track almost perfectly with the incidence of influenza virus. So if this new virus behaves as old coronaviruses do, then we might expect the same sort of trend. Now, I will say that SARS, which occurred in 2002, 2003, began in November, peaked in the winter, and was gone by early summer. So it behaves in a very similar pattern. That one did not recur ever yet, anyway, and hopefully never. So I suppose that's the best case scenario for this new virus. What do we know about coronavirus immunity? Well, unfortunately, it turns out that infections with the common coronaviruses recur. So you might get infected this year, and a couple more years later, you very well may get infected with the very same strain of human coronavirus. And this can occur repeatedly. And probably the reason for this is that the antibody response, while protective, is protective for a fairly short period of time. And then the antibody protection wanes and you become again susceptible. And this also seemed, interestingly enough, to be the case with SARS. So if you follow patients who were infected with SARS in 2002 or 2003, over a 3-year period, you can see that their antibody responses peak at about 90 days, about 3 to maybe 6 months. And then they decline fairly rapidly, such that by about 3 years after the time of infection, antibody titers are rather low. Now, this does not mean that these people will not be protected if they came in contact with the SARS virus again, because they may, for example, develop an amnestic response, or they may have T cells that can protect them from subsequent exposure. We don't know, and hopefully we'll never find out. But anyway, this certainly gives us pause when we think about this new virus, whether immunity will be lasting or not. What we do know about the antibody responses to this most recent outbreak is that they seem to follow a pattern that you would see with any other viral infection, where you first get an IgM response, and then maybe a few days later start seeing IgG. And we would expect the IgM to wane fairly rapidly over the course of a few weeks to a month. And what we don't know, though, is whether the IgG antibodies will stay elevated, and if they do, for how long. There is a little bit of good news. It's a tiny bit of good news on this front, I have to say. Somebody did an experiment where they infected monkeys with SARS CoV-2. These monkeys recovered. They then waited a month and they reinfected them, and they could not infect them. So they seem to be immune following infection with SARS CoV-2. So that's good news. The bad news there, though, is that, first of all, there were only 2 or 3 monkeys that were in the study. But probably, more difficult to get your head around is the fact that these animals were challenged only 28 days after infection. So what you can say from this data is that if the monkey data apply to humans, and you get exposed 28 days, unluckily after your last infection with COVID, that you're probably going to be protected. But we can't say that if you get exposed 6 months later or a year later, they're not going to be protected. Now, there are these anecdotal reports of recurrent COVID-19 infections. These are probably erroneous and are probably due to false negative tests, where somebody had a positive test, maybe another positive test, then they had a negative test, then another negative test, and then maybe a positive test after that. Chances are those negative tests were false positives. Sorry, false negatives. And that these people really were just shedding virus for prolonged periods of time. What about vaccines? Well, there are several that have been tested in animal models against the MERS and the SARS agents, and these have been effective. They come in various formats: inactivated vaccines, DNA vaccines, subunit protein vaccines, and then vaccines that are vectored in by various viruses, particularly adenovirus. So that's very hopeful. And in fact, there has been a phase 1 human trial of a DNA vaccine against SARS in humans. It was shown to be immunogenic and safe. It was, of course, never tested for efficacy because there were no further cases of SARS. I think many of us have heard about the new vaccine that's just been rolled out. This is being sponsored by the National Institutes of Health and a company called Moderna. This is a vaccine that is made of messenger RNA that expresses the spike protein of SARS CoV-2. There's some real advantages using mRNA vaccine, the main one being that it's very easy to make. You only have to synthesize it. You don't have to grow it in cells or in eggs like you do for influenza virus vaccines and others. There are a couple other vaccines that are being rolled out, one in China that's based on adenovirus, and another mRNA vaccine being rolled out by a German company. So, obviously this is something that'll be very interesting to follow. We're a ways away, unfortunately, from both knowing whether this is safe and effective, and of course distributing it widely. We don't know a lot about the pathophysiology of COVID-19 based on SARS, which seems to have probably a similar pathophysiology. We think that it's mainly the pneumocytes, particularly the type 2 pneumocytes, that the primary target of infection in the lung. Alveolar macrophages seem to get infected, though less commonly. There appear to be macrophages and neutrophils that infiltrate in response to this infection and probably set up immunopathology, which is characterized by pro-inflammatory cytokines such as IL-6, tumor necrosis factor alpha, and interleukin-8. Interestingly, there is a delayed interferon response, but it persists. This is the reason this is delayed, because very early on in infection, it's nice to have interferon. These viruses are probably sensitive to interferon. They devote a lot of their energy to inhibiting interferon responses early on, and so they manage to evade that response. However, because of the pro-inflammatory responses that these viruses engender, there becomes a delayed interferon response that persists and probably causes more harm than good. Some of the damage, phagmosa damage, is seen at the level of the alveoli, and eventually one winds up with an ARDS and a fibrinous organizing pneumonia. And I was able to find at least one paper just out showing some of the pathological features of COVID-19. This was fortunately an autopsy case. And what you see is diffuse alveolar damage, similar to what you would see with SARS. There's edema, there's hyaline membrane formation, as you can see in ARDS and mononuclear infiltrates. We do not want to get to this point. This was an autopsy. We want to treat our patients and prevent this. And that's a good segue to our next speaker, Dr. Shruti Gohel, who will talk about clinical manifestations of this infection and how to hopefully prevent this scenario.